Welcome back. What we're going to do in this video, and this is probably going to be sort of a lengthy video, but this video is going to be on something called receptor guanylyl cyclases. Okay? So in this video, we're going to explore the various hormones and things like that that activate guanylyl cyclase, but also we're going to explore mechanisms as to how they work. And what I really want to start doing in these videos, I haven't really stress this up at this point, is that I want to give you applications as to why this is important. So it's not just memorizing things. Okay, so if you're a doctor, when might you find this being important? If you're a kinesiology person, when will you see this in practice? Things like that. Okay, so this particular video is going to be on guanylate cyclase. Okay. Now, there are two main molecules we're concerned with. Okay, we're concerned with something called ANP and nitric oxide. So ANP is called atrial natriuretic peptide. Okay, sometimes you'll see it written as ANF. That just means atrial natriuretic factor. They're the same thing. And then, of course, you have nitric oxide. Okay, now before we get into the nitty-gritty details, let's talk about the general function of both of these. Okay. So atrial natriuretic peptide, at least that's with the peptide, that's the way I always learned it. But what it's effectively doing is it's increasing the excretion of sodium and water at the collecting ducts in your nephron. So ANP is going to act at the nephron, and it's going to cause the excretion of sodium. And because water typically follows salt, whenever you lose the sodium due to osmotic changes, water leaves as well. Okay, so effectively what ANP does is it's going to lower blood volume, and consequently that lowers blood pressure. So it turns out that atrial natriuretic peptide is synthesized and released by the right atria in the heart. And there's actually receptors in there that monitor the blood pressure, so when the blood pressure rises too much, ANP gets released and this process occurs. Okay, so it's a way of negative feedback regulation. Okay, so in other words, ANP is lowering the blood pressure. Okay, nitric oxide does a very similar thing. It ends up lowering the blood pressure, but it does so by vasodilating. So the ultimate result of increased nitric oxide secretion is vasodilation. Okay. Vasodilation. So what ends up happening is you increase the blood vessel diameter, and that also decreases the blood pressure in that area. Okay. So blood pressure goes down. Blood flow also increases. So one thing that's also important is you increase blood. We'll actually write it out because it's so important. Is that you'll increase blood flow. Okay. So the increased blood flow, what we'll see is that it actually leads to some of the important properties of it. Okay, but let's actually talk about the receptors. Okay, for these particular molecules. So first thing we'll talk about ANP. Okay, so we'll draw a little cell membrane here. So here's our cell membrane. Okay, and in the membrane we're going to have a homodimer. Okay, we're going to have a homodimer. Okay. And the homodimer is going to look something like this. And do my best. It doesn't have to be exact, but So you have a homodimer of these guanylate cyclase units. Okay. Specifically, the guanylate cyclase activity is right here. Okay. So if this is our cytosolic side, this is our cytosolic side. Out here's the extracellular fluid. Right. So it turns out that the catalytic side of it is the cytosolic side, the intracellular side. Okay. So what's going to happen is you're going to have ANF that's going to bind in here. It's going to bind somewhere in a pocket around here. So atrial natriuretic factor or atrial natriuretic peptide binds here, and that induces a change in conformation of the protein, okay, the receptor. That change in conformation is spread from the extracellular side through the transmembrane domain into the intracellular side, okay? And when that occurs, you get activation of the catalytic side of the cyclic GMP receptor, okay? So the actual receptor side is out here, right? And the catalytic side is on the inside. And specifically, the inside is going to be your actually guanylate cyclase domain, okay? So what's going to happen is it's going to take GTP and it's going to convert it into cyclic GMP, okay? Let's actually look at how that happens, okay? So let's go ahead and draw GTP, okay? So what you're going to find about this particular mechanism is if you watch the mechanism if you've watched the mechanism on adenylate cyclase, what you'll find is that this mechanism is virtually identical. Okay. In fact, the actual organic mechanism, with the exception of the fact that you're dealing with guanine, okay, the mechanism is identical. Okay. So what we're going to have is we're going to have our imidazole-like ring here. Here's our imidazole-like ring. And let's see, you have this right there, this there, and this is a proton. Okay, there's GTP. Okay, so effectively what guanylate cyclase is going to do is it's going to catalyze a nucleophilic attack. It's going to be a nucleophilic acyl substitution. Okay, so this is our 3' hydroxyl group, right? This is our 3' hydroxyl group. Again, this is our 5' triphosphate. Okay, so the 3' hydroxyl group is a nucleophilic attack on the alpha phosphate. The electrons kick up, generate a trigonal bipyramidal intermediate. The electrons kick back down, and they force off the leaving group, which in this case is going to be pyrophosphate. Okay, and what you'll find is that there's a nearby inorganic pyrophosphatase that drives the reaction by producing 2 phosphates with a hydrolysis. You get a release of free energy, Gibbs free energy, which drives the reaction forward. Okay, so what do you get? Okay, this reaction, by the way, is irreversible under normal conditions. Well, I say normal, I mean it is irreversible under physiological conditions. Okay, so what you end up with is something that looks like this. Okay, so you still have your guanine nitrogenous base here. It's pretty poorly drawn there, but you get the point. Okay, so here's your guanine guanine nitrogenous base. Now what ends up happening is, so what you get is you get this cyclization, right, that looks something like this. Okay, and effectively what this is cyclic GMP. Now, much in the same way we name cyclic AMP, this has a special name. Okay, remember that this is the 3' carbon and this is the 5' carbon. So the way we name this is 3', 5' cyclic GMP. You could also call it 3', 5' cycloguanylate. That will also work, but we typically just call it 3', 5' cyclic GMP. Although because overwhelmingly the main type is 3', 5', we usually just reduce it to cyclic GMP, much in the same way we did with cyclic AMP. Okay, and just remember that we have a loss of pyrophosphate here, right? And that the pyrophosphatase drives the reaction with the release of free energy. It's a hydrolysis, and you end up getting 2 inorganic phosphate. Okay, again, it's a hydrolase. Okay, and that's what happens when atrial natriuretic peptide binds to its receptor. Okay, you have a homodimer of receptor guanylate cyclases, and when ANF binds, the actual catalytic activity of the guanylate cyclase becomes activated, and it catalyzes the irreversible conversion of GTP to cyclic GMP. Again, it's an intramolecular nucleophilic acyl substitution, and of course, this guy is your final product, cyclic GMP. Okay, so how does ANF do what it does? Well, it turns out that the cells that contain the collecting ducts, okay, in the kidney, they specifically bind cyclic GMP. Okay, and when you have that rise in cyclic GMP, it triggers the excretion of sodium, and of course, following with that is your water. Okay, so cyclic GMP binds to receptors. Okay, and ultimately, what it induces is excretion of sodium and excretion of water. Okay. So that's your ANF mechanism. Okay. Now you're going to have a similar thing for your nitric oxide, except for the fact that it's not membrane bound. Okay. Instead, let's carry out our membrane a little bit further here. Okay. Instead, with our nitric oxide synthase, not nitric oxide synthase. Instead, our guanylate cyclase is going to be cytosolic. Okay. So we have a cytosolic enzyme, okay, and specifically it has a heme, okay, the heme here, okay, so that was terribly drawn. Let me do it like this. Oops. let me do it like this. So if I have my iron in the center, again, you know from the hemoglobin videos, you have your coordination of the iron through the pyrrole nitrogens, okay? So just know that this is soluble guanylate cyclase, okay? Now what ends up happening is nitric oxide is going to bind to this heme, okay? So you're going to get nitric oxide that binds here, okay? And when it binds to the heme, there's a change in conformation. Again, recall that the heme is going to be attached to the protein itself. So, and I believe specifically it's attached through a histidine residue. So you can imagine this is the imidazole nitrogen of the histidine. And when the iron moves, the histidine moves. And remember our thing from hemoglobin, you know, the foot bone's attached to the ankle bone, which is attached to the shin bone, which is attached to the leg bone and all that stuff. When the heme moves, the histidine moves, and so the whole protein moves. So there's a change in conformation when nitric oxide binds, okay? And by the way, I don't know if I mentioned it, but you have an enzyme called nitric oxide synthase, okay? So effectively, when you need nitric oxide, that enzyme basically catalyzes the irreversible conversion of arginine to nitric oxide. So effectively what you have, let me just go ahead and draw it. What you're going to have is you're going to have arginine. So here's going to be arginine, okay? So 1, 2, 3. So yeah, you're going to have arginine, and there's going to be an irreversible conversion of it to nitric oxide. And you're actually going to have a fairly large leaving group in this case. Your leaving group is going to be citrulline, okay? So what you're going to end up with is this. Come ahead and draw it over here. So effectively what you have is arginine gets converted to nitric oxide, okay? So this is arginine right here. Okay, this is arginine. If you haven't seen the urea cycle yet, this molecule right here is citrulline. It's an amino acid, so we call it L-citrulline, right? But effectively, your leaving group is going to be nitric oxide. So nitric oxide is going to come over here, and it's going to bind to the heme, okay? And what this does is, again, it changes the conformation of the soluble guanylate cyclase. And so you get the cyclization of GTP. So GTP comes in here, and you end up with this guy right here. Actually, it's actually this down here. Sorry. You end up with GMP. So the point is that GTP cyclizes to cyclic GMP. So in either case, whether you're talking about ANF or nitric oxide, you're going to get a rise in intracellular cycloguanylate, cyclic GMP. But it does so through a different mechanism. In the case of atrial natriuretic factor, right, that's a homodimer in the membrane. And so the actual ANF binds extracellularly, right? And so you get rise in cyclic GMP through the actual receptor cytosolic catalytic protein, right? In the case of nitric oxide, you have intracellular enzymes, which I believe are microsomal. They are they're oxygenases, they're nitric oxide synthases, and they catalyze the irreversible conversion of arginine to L-citrulline and nitric oxide. Nitric oxide binds to the heme on this soluble guanylate cyclase, changing conformation, and it cyclizes GTP to cyclic GMP. Okay? So that's good and all, but why would this ever be important? Well, anytime you want vasodilation, you have nitric oxide synthase. Okay, so this, by the way, this enzyme right here is nitric oxide synthase. Okay, so anytime you need vasodilation, you have to have nitric oxide synthase, right? So let's come down here and let's think of times you might want vasodilation. So you're going to want vasodilation usually when you activate the sympathetic nervous system, right? Well, excuse me, the parasympathetic nervous system. That was very stupid of me. Okay. Anytime you have relaxation, right? If you're sitting at your computer right now, your blood vessels are dilated, right? So they're going to have nitric oxide in them, right? If you look at healthy cardiovascular physiology, your blood vessels at rest in healthy conditions, they have nitric oxide being synthesized. And it's synthesized by the endothelial cells. Anytime you want to maximize blood flow and you want vasodilation, that's the action of nitric oxide synthase, among other things. Okay? So one area where this is applicable is human sexuality. Okay? So one thing that we can consider is the human erection. Okay, and generally in society and common talk, we normally just think of it in terms of males, although females have it as well. The male penis is embryonically homologous to the female clitoris. They're both of them. The penis and the clitoris are derived from the same embryonic tissue, so you'd expect them to behave similarly, even though they look different. So when a male or female gets sexually excited, that corresponding genital region increases blood flow, and it does so through nitric oxide. Okay, so nitric oxide gets synthesized by those cells in the blood vessel walls, and you end up getting increased blood flow, and that's what facilitates the erection in both males and females. Okay, but what happens when that blood flow is limited? What happens when blood flow is blocked? And in the context of American diets, that's a very common thing. Normally, where we see things like erectile dysfunction, which, as they say in the commercials, is caused by lack of blood flow, is normally not really so much of an age thing. It's more of a poor diet thing. There are lots of things that really include things like high sugar in the diet. These kind of things can facilitate blockage of blood, usually through clogged arteries for things like plaques in the arteries, things that lead to the penis itself. These are the kind of things that promote loss of blood flow. Okay, in other words, blockage of it, so that you can't get to the blood to the capillaries. Therefore, blood flow is minimized. Okay, so ordinarily, in a healthy individual, the individual should have no problem getting blood flow through their genitals, right? But what we have to also consider along the side of your guanylate cyclase with the cyclic GMP is that there's another enzyme working as well, and this enzyme is called cyclic GMP phosphodiesterase. Okay, and for actually this, I'm actually going to go ahead and use the structure I already had drawn because it'll be easier. Okay, what is guanylate? What is cycloguanylate phosphodiesterase? So what's a phosphodiesterase? Well, phosphoesters, right? This group right here, by the way, that I'm about to circle, this is a phosphodiester. So if you have a phosphodiesterase, what do you think it does? Well, it's just going to hydrolyze the bond, right? So what you're going to get with the phosphodiesterase is water is going to come in, right? And it's going to do a hydrolysis. So nucleophilic attack on this phosphorus. These electrons kick up, generate a trigonal bipyramidal intermediate, right? The electrons are going to kick back down and kick off your leaving group, which in this case is going to be the 3' hydroxyl group, which will come out and abstract a proton. So what you end up getting, and I'm going to come over here and draw it, just for the sake of space, what you end up getting is something that looks like this. Okay, so again, you regenerate your 3' hydroxyl group, and you're going to keep your guanine nitrogenous base, right? So that stays the same. But what ends up happening is now your phosphate is no longer in a phosphodiester bond. It's now simply a 5' phosphate. So this is your 5' carbon, right? So it's a 5' phosphate. So we call this 5' GMP. Okay, it's no longer cyclic, it's just GMP, right? This is sort of the degradation product of your phosphodiesterase. Recall that when we did the actual regulation of your adenylate cyclase, you had a corresponding adenylate, cycloadenylate phosphodiesterase. Its mechanism is identical to this one, right? It's just a simple hydrolysis at the cyclic phosphate, right? Loss of the leaving group, which is your 3' hydroxyl group. Okay, so you have a corresponding competition between your rise in intracellular cyclic GMP and the phosphodiesterase. So if you want, let's say you want to promote an erection, how would you do that? Well, you would have to think, okay, well, if I want to keep the erection, I'd want to have my cyclic, conceptually at least from that standpoint, you'd want your cyclic GMP levels to outweigh the actual degradation of them, right? So in other words, you'd want to have enough cyclic GMP so that your phosphodiesterase doesn't eliminate it all, right? Because if you think about it, if the phosphodiesterase is getting rid of cyclic GMP, right, what's it doing? Well, cyclic GMP promoted the erection, right, by promoting vasodilation. So if phosphodiesterase breaks down cyclic GMP, then therefore the phosphodiesterase inhibits the human erection in both males and females. Okay, so what's the problem in someone with, someone who has erectile dysfunction? Is it that they have too much activity of a phosphodiesterase? No, it's that they have blockage of blood flow. So even in the presence of nitric oxide, they have limited blood flow because they have some sort of clogging of their arteries or their arterials or whatever you're talking about that leads to the penis, right? So there's some blockage, okay? So effectively what we're going to look at right now is a molecule, and I'm going to try to draw it correctly, okay? It's going to look very similar. It's going to look very similar to guanylate, okay? And you'll see that it almost looks identical in one area, okay? This molecule is going to be Viagra, okay? The actual correct name of it is going to be sildenafil, okay? Oops. That is supposed to be a single bond, okay? And what we're going to do is we're going to sort of analyze this structure, okay? So what I want you to notice, first of all, is this group right here that I'm drawing, okay? Notice, what does this look an awful lot like? Well, actually, let me go ahead and finish drawing the whole structure, and then we'll come back to that. So there's an aromatic ring. Here's your hydroxyethyl group. Then you're going to have this sulfate group right here. And that's going to be attached to this very strange group right here. Okay? This molecule right here is called sildenafil. Sildenafil, it's marketed as Viagra. And the original function of this drug, most people don't realize this, it was actually a medication for angina pectoris. So if you had thinner blood vessels, in other words, you had blockage of flow, you needed to dilate, right? Doctors would give you this drug, sildenafil, and what it would effectively do is widen your blood vessels to drop your blood pressure, right? Lowers blood pressure by widening the diameter of the blood vessels. So any kind of constriction you had, or basically the point is that it maximized blood flow, okay? And it promoted that in a healthy manner, okay? And actually, let me go ahead and do this because I actually misdrew part of the structure. This should actually be part of the ring. But anyways, okay. But basically, it was marketed for angina pectoris. So you give someone sildenafil, and it widens their blood vessels so that they maximize their blood flow. Now, it did so through a cyclic GMP mechanism, but it doesn't actually increase cyclic GMP. So how does it work? Well, it turns out that this molecule, notice this part of the molecule right here. What does this look very similar to? Well, it looks similar to guanine, right? And it turns out that this particular molecule can fit in the active site of the cyclic guanylate phosphodiesterase, right? And it turns out that this molecule is a competitive inhibitor, okay? So if you're inhibiting the phosphodiesterase, your cyclic GMP phosphodiesterase can't degrade cyclic GMP. So you have more cyclic GMP for a longer period of time. More cyclic GMP means vasodilation, more vasodilation, and that means that you'll potentially be able to maintain an erection for a longer period of time. So this guy right here is sildenafil. So let me go ahead and rewrite that because I botched the structure originally. This is sildenafil, and it's, again, marketed Viagra. And again, it fits into the active site of the phosphodiesterase and inhibits it, okay? So you give this to someone and it maximizes blood flow, okay? Now, they often say with sildenafil when you're taking it that you have to watch out for things like vision problems, hearing problems. The reason for that is because those biosignaling pathways also tend to use cyclic GMP as well. So if you have cyclic GMP mechanisms there, you probably also have a cyclic GMP phosphodiesterase there as well. So if you have a ways to synthesize it, you're going to have a way to catabolize it, okay? So that can potentially pose problems for vision and hearing because they have a phosphodiesterase as well, but for the purposes of maximizing blood flow in the human genitals, this is the drug you want to use, okay? So let's do a quick recap of what we've seen, okay? Let's talk about atrial natriuretic factor first. This is a hormone that gets released from the right atrium of the heart, and it binds to homodimer receptors, which are guanylyl cyclase receptors. The actual receptor part is on the extracellular side. Excuse me, when it binds, there's a change in conformation, and you get activation of the guanylate cyclase on the intracellular side, and GTP gets cyclized to cyclic GMP. Okay, it's in this mechanism right here. You have GTP, right? There's an intramolecular nucleophilic acyl substitution on the alpha phosphorus. You get a loss of pyrophosphate. A pyrophosphatase nearby drives the reaction forward. You get a release of free energy. The exergonic nature of that tends to drive the reaction forward, right? Release of free energy. And you get this guy right here. This is your cyclic GMP. Specifically, it's 3', 5' cyclic GMP, right? And essentially what that does in the case of ANF is it binds to receptors and promotes the excretion of sodium by the collecting ducts of the kidneys. And consequently, as you lose sodium, you also lose water. Okay, that's good and all. Now what about your nitric oxide? Well, recall that we have this enzyme called nitric oxide synthase. Like I said, I believe it's part of the endoplasmic reticulum. Okay, and it takes arginine and it catalyzes the irreversible removal of L-citrulline with your loss of nitric oxide. Nitric oxide comes up here and binds to this heme, right? And remember, the heme is coordinated by histidine on the axial position in the case of your soluble nitric oxide or soluble guanylate cyclase, right? And remember, as soon as nitric oxide binds, it's like, remember, the foot bone's connected to the ankle bone and so forth. When you bind nitric oxide, the heme moves, so the histidine moves, so everything moves, and there's a change in conformation. And effectively, GTP gets cyclized to cyclic GMP, okay, through the same mechanism that we've seen. Cyclic GMP then binds to receptors on the endothelial cells of the blood vessels, and it promotes vasodilation. Okay, now why is this important in practice? Well, remember that we have this enzyme called a phosphodiesterase. So here's our phosphodiesterase. And phosphodiesterase, all they do is they basically hydrolyze phosphodiester bonds, right? In fact, if you degrade nucleic acids, you're actually using phosphodiesterases. So things like DNases, those are phosphodiesterases. However, these ones are specific for cyclic mononucleotides, right? So this is going to be specific for cyclic GMP. And so it hydrolyzes cyclic GMP in the mechanisms that we saw for cyclic AMP catabolism, right? Hydrolysis of the phosphorus, right? Hydrolysis of the phosphate, attack on the phosphorus, loss of the 3' hydroxyl group as your leaving group, and you end up with 5' GMP. Okay? 5' GMP has no vasodilator activity. Only cyclic GMP does. So if you terminate cyclic GMP activity through phosphodiesterase, you're effectively lowering vasodilation. So people with erectile dysfunction don't have a higher level of phosphodiesterase. No, they just have blocked arteries, so it makes it more difficult, even in the presence of nitric oxide, to maintain an erection, right? Because you have blockage of blood flow. So what sildenafil does is it mimics the structure of GMP, or cyclic GMP. Here's the guanylate component, right? Or the guanine, I should say, right? So this resembles guanine, right? And it gets into the active side of the phosphodiesterase and acts as a competitive inhibitor and blocks it, right? So the phosphodiesterase can't function, okay? So it prevents the hydrolysis of cyclic GMP, so it remains there longer. So you get more of a vasodilator effect by having cyclic GMP there longer, okay? And so as long as that sildenafil is there, you get the blockage of the phosphodiesterase, and effectively you can maintain the erection of both the penis and the clitoris. Remember that embryonically they're derived from the same tissue, so they have the exact same function, the same types of receptors, the same physiology, and all that. Okay, so this is your general function of your cyclic GMP receptors. Okay, one important thing I want to point out, if it wasn't apparent at this point, is that in ANF, remember, it's a membrane-bound receptor. The receptor component is part of the catalytic component. They're part of the same protein. It's just that the receptor component is extracellular, and the catalytic component, GTP to cyclic GMP, is cytosolic. Okay? And, of course, it's a transmembrane domain. And in nitric oxide, it's a soluble receptor. Okay? And in that case, cyclic GMP synthase activity is going to be part of the soluble protein itself. Okay, so I hope this video gave you a little bit of intuition on what's happening with cyclic GMP. See you in the next video.